Well, thank you, everybody, and good afternoon. My name is Chris Nevash. I am the Senior Product Manager for Gusmer Enterprises, and I'd like to thank you for your time today in joining Gusmer and Christian Hansen for our webinar on malolactic bacteria. On that note, I'd like to roll this over to David Spector, who is with our industry partner from Christian Hansen. David?
Hello, everyone. Thank you, Chris, and thank you all for joining our webinar on malolactic fermentation. My name is David Spector, and I'm joining you from Venice Beach. We also have my colleague, Duncan Ham, who is joining us today from New Zealand. And I'll just take 30 seconds to introduce Christian Hansen. We're a global biotech company with the world's largest library of bacteria. And you're probably joining us today because you know Christian Hansen is the industry standard when it comes to Oenococcus oeni for malolactic fermentation. Now, we all know environmental yeast and bacteria can carry out alcoholic and malolactic fermentation. And this certainly can yield great results. You know, some of the best wines in the world are made this way. But you must deeply understand your vineyard and your winery's microflora if you don't want to leave the output of your wines up to chance. And so what we're going to talk about, mostly what we're going to talk about is control. Control over your process by understanding the microorganisms that you're dealing with, how to optimize their performance, and then using commercial strains to help you achieve your goals. Next slide. Today, we're just going to focus in on Oenococcus oeni and mastering your malolactic fermentation, but I do want to highlight that we at Christian Hansen and Gusmer believe that malolactic fermentation is just one part of the fermentation process, and we suggest attending to each step: pre-alcoholic fermentation, alcoholic fermentation with precision and with care. Next slide. So this is the classic sequential inoculation sequence in winemaking. We try to avoid taking a reductionist approach to winemaking. So rather than just focusing in on one element in isolation, I always like to start off with the fermentation process holistically. And so with this graphic, you know, we're overstressing the rise and the fall of certain microbiological populations, but it really allows us to begin the conversation on inoculation timing, which Duncan is going to explore in more detail during this presentation. Next slide. I also wanted to show you our entire range of Oenococcus oeni for malolactic fermentation. You'll see this slide a couple of times throughout. And you know, having a wide range of strains, each with unique properties, really allows the winemaker to achieve their goals. Perhaps you have a wine with high ethanol. You might want to look at CH16, or you want the stability of malolactic fermentation, but not the diacetyl. You probably want to take a look at Cine. So you really will work together with your technical representative at Gusmer to pick the right strain for your wine. Next slide. So, you know, I really hope that today is just the start of the conversation on improving your control, optimizing your malolactic fermentation protocols. You know, our partner and North American distributor Gusmer has a ton of resources available on their website, in their catalog, and they have plenty of technical experts to help you. So you're certainly not in it, you know, by yourself. You take this information and go with it. No, this is definitely a collaborative process, and we hope to work with you on all of your unique wines. Okay, so now I'm going to get to the part you all came for, and that is to introduce Duncan Ham, who will be leading the bulk of this presentation. So Duncan Ham is currently the regional technical manager for the Asia Pacific region, based out of Christian Hansen, Australia. But pretty soon he's coming to our headquarters, our Christian Hansen headquarters in Denmark, where he's going to work as our senior application scientist. So hopefully, if you have some trials lined up with Gusmer, maybe you'll be seeing Duncan come out to North America and helping you with those trials. Duncan studied chemistry at the University of Waikato in New Zealand. He's going to have to tell me if I pronounced that correctly. And then he's spent a lot of time, 10 years in the dairy industry, including 5 years with Christian Hansen. And that's certainly how Christian Hansen got into the bacteria business, bacteria for cheeses, for yogurt, sour creams. And so after completing a graduate diploma in viticulture and enology at Lincoln University, he got some hands-on cellar experience and has been working in our enology department ever since. So yeah, he's looking forward to working with us here in North America. And without further ado, I'll let Duncan take over the presentation now. Welcome, Duncan.
Thank you, David. Thank you very much. And just your pronunciation of Waikato is pretty close, I have to say. So, yep, it was pretty close. So thanks a lot for that really kind introduction. And really looking forward to the next 30 minutes or so to talk about mastering malolactic fermentation. And really hope that what we are presenting today is useful and constructive. So I thought it'd be really good to start with just a few definitions and an introduction to what malolactic fermentation is. It's an enzymatic conversion of L-malic acid, which is naturally present in grape juice and wine, and it's a conversion of that acid to L-lactic acid. There's some debate as to whether it's actually a fermentation. Traditionally, it was just seen as a conversion. I think the scientific community today would generally agree that it is a fermentation, but nevertheless, there is this transformation. Now, there's a reason why the bacteria that do malo, and that is it allows them to survive and grow. So it allows them to derive energy in the form of ATP from ADP, and it helps them to flourish. Now, what does it actually do to the wine? Malolactic does soften the acidic profile, and it does this because it both increases the pH, but it's also because the nature of lactic acid tastes the taste of lactic acid is much softer than that of lactic acid. And there's a good chemical reason why. Malic acid is diprotic. It contains 2 carboxyl groups. Lactic acid contains 1. So it's actually less acidic by nature. But malo doesn't just soften acidity. It will also affect the aromatic properties of the wine. And it can be in various ways. It could be through the production of diacetyl, which gives that buttery flavor, or it could be through modulating the fruit impact as well. And lastly, and this is a particularly traditional European idea around malo, and that is it gives microbial stability. So back in the days before sterile filtration and the level of hygiene that we have in modern wineries, by doing malo, you're effectively removing a fermentable substrate from the wine, so you're less likely to get malo happening in bottle, which effectively is a form of spoilage. Now, all of this happens in a very, very harsh environment. Wine not only has an exceedingly low pH, but also has a high level of alcohol. So it's a pretty toxic environment to pretty much almost all microorganisms. However, certain very robust bacteria can survive and can flourish in wine. So when we do talk about malo, we tend to talk about Oenococcus oeni as a predominant strain that will drive this. It's not the only species that can do it, but in most wine conditions, it's the only species that will survive well enough to do it. But as we mentioned, wine is an extremely harsh environment, and the 4 main stress factors would be the level of sulfites, SO2, the pH of the wine, the concentration of alcohol or ethanol, and also the temperature. Now, all of these stress factors tend to work synergistically, meaning that if you're perhaps high alcohol, it's going to be much higher to ferment something with a very, very low pH and a high level of sulfites. So they tend to compound on one another. And I'll just make a comment about temperature. Temperature we talk about as a stress factor. Lower temperatures are not necessarily bad for bacterial survival, but with bacteria being very, very simple organisms, they rely on the heat of their environment for their metabolic pathways to work. So if it is too cold, they won't ferment. The flip side, if the temperature is too warm, that will actually, if it's a wine, it will actually kill the Oenococcus, because above around 25 to 28 degrees, Oenococcus will start to bring alcohol across the cell wall, which will be toxic to it. Now, I just also wanted to mention a few other stress factors, which we'd probably refer to as secondary stress factors. One of them is low nitrogen. Malo requires around 60 parts per million of amino nitrogen in the wine for it to proceed. And one other interesting facet we see in some wines, particularly ones that are fermented with champagne style yeast, is the presence of medium chain fatty acids. These are derived as a stress response from some of these Saccharomyces strains. However, they are very toxic to bacteria, but they can also be dealt with quite effectively by using yeast hulls or malo nutrients, and I'll talk about that further down the line. Just one other comment. I sometimes see press saying that a high level of malic acid is actually bad for bacteria. We would actually dispute that. Actually, having a high concentration of malic acid can be a really good thing for malo. The really important factor is the pH. Often wines that do have high malic levels do have a low pH, but not exclusively so. And we've seen plenty of examples of wines with 5 or 6 grams of malic acid, but a moderate pH, have absolutely really successful, clean and fast malolactic fermentation. And that's simply because malic acid is a substrate for the cells. And if you have quite a lot of it, that's not a bad thing. So a bigger problem is when you drop below about 1 gram, it becomes far more difficult. So to talk further about the stressful environment of wine, we've just thought we'd break it down into those 4 primary factors. So first of all, with pH, we thought about we'd try and put some numbers behind what's difficult and what's favorable. So generally, wine below pH 3, we would class as difficult. That's very difficult. These wines often require quite a lot of work to go through. It's not impossible to get them through malo, but it does take work, often maybe adapting a population of bacteria, or just giving the wine a long, long time and making sure all the other factors such as sulfites and alcohol are not extreme. 3.0 to 3.2 pH tends to be a little bit difficult, but we have direct inoculation cultures which will ferment within that range. But the most favorable tends to be around 3.2 to 3.7. Above pH 3.7, we're talking very favorable conditions, but that also carries a risk with it because it also becomes more favorable for spoilage organisms. But fortunately, inoculating with a high level of malolactic bacteria is one of the most effective things you can do to help prevent things like Brettanomyces or Pediococcus growing in your wine, because you're dominating the must with a good, clean strain. The second point is temperature. Below 59 degrees Fahrenheit, we tend to see malo as either very, very slow or generally non-existent. Between 59 and 66, it can proceed, and we have strains that will proceed at this level. But really, the best range is to be between 66 and 77. Above 77, we start to see a slowdown again, and around 85 Fahrenheit, we start to see cell death. And as I mentioned earlier, that's because Oenococcus can actually tolerate higher temperatures without the presence of alcohol, but once you have alcohol there, it comes across the cell wall too easily and will actually poison the organism. So it's important to keep the wine below 77 Fahrenheit. The next point is SO2. Obviously, SO2 is inhibitory to microorganisms. That's one of the reasons why it is used. But in terms of numbers, above 45 parts per million, we'd say is really, really difficult. We do see wines go through malo at this, above this from time to time with specific strains. But if it can be avoided to be that high, then it's really strongly recommended to do so. 30 to 45 ppm isn't so bad. Some strains will start to struggle more than others in this range. 15 to 30 ppm is less favorable. And then as the lower we go, the easier it becomes for the malo bugs. And so below 15 parts per million is certainly what we would regard as favorable. The last point is ethanol. So below 13% ethanol should not pose any problems. We would start to see some difficulty between 13 and 15%. At this point, it would be important to start looking at a strain that has a high degree of alcohol tolerance. And 15 to 17% is where you really need to use a specific strain to get the wine through malo, because a lot of Oenococcus will not survive at those sort of levels. So I'm going to move on now to the different approaches to getting a population of Oenococcus into a wine. And in Christian Hansen, we would say there's 6 ways you can do this. Just 6, no more. The top one we've highlighted here is to rely on spontaneous flora, and that would be lactic acid bacteria that's either come through on the skin of the grapes and been living through the wine and the fermentation process, or has somehow naturally come into the wine through the winery environment. The second point is cross-seeding. This is where you would take an active ferment, which will have an active population of Oenococcus, and move it across to a tank that you'd want to inoculate at somewhere between 1 and 5%. Then the next 4 that we've got here all are around cultures. We have build-up cultures, which take somewhere between days and weeks to propagate and build up in different stages. Then we have pre-activation and quick reactivation. These are similar types of cultures. Pre-activation ones require preparing them around 24 hours before you want to use them. And quick reactivation, it's around 30 minutes before. I like to think of those as probably being akin to rehydrating an active dried yeast. It's that sort of level of work, although with the pre-activation ones, it does take a bit longer than the quick reactivation. But nevertheless, there is some preparation required. And then lastly, we have direct inoculation. So all 4 of those are commercial products that you would buy. So they're commercial cultures. But as we go through the list from build up through to direct inoculation, we'd say there's actually an increase in the perceived cost. And I say perceived rather than actual cost, because the flip side of that is that there's more risk involved with the more basic cultures. So the more advanced the cultures, the less risk. And actually the costs arguably are lower using something like direct inoculation. It's the highest quality cultures you can buy. And in Christian Hansen, we only produce direct inoculation cultures. The direct inoculation works around 2 key facets. The first one is getting a really high number of cells. And I'm going to talk further about what we call the magic number. But it's about having that critical mass for malo to happen. But you don't just want to have a high level of cells. The other really important thing is to have cells that are really well adapted. So in our production process, we make sure that the Oenococcus cells are pre-adapted to wine. So when you do inoculate them, 100% of them will survive and won't die, and we'll be able to start the job within days. Just to talk about spontaneous malo, and David made a really good comment earlier that some of the world's most famous and iconic wines are still undergo spontaneous malo, and it's where the malolactic happens, and it's carried out by adventitious flora. Now, this flora could come through from the vineyard, it could be in the winery environment, it could be on the equipment, but it's a case of waiting for this level of this flora to inoculate the wine and to build up. It's still used quite widely in many regions, although over the last 20 years, there's certainly an overall trend or shift towards inoculation. And there's perception from winemakers that the culture is free, and that's a valid approach. And I've heard on numerous occasions winemakers say to me, as someone who works for a culture company, why buy something that I can get for free? And that's true. You don't pay for the culture, but the flip side of that is that there's a lot more risk involved with spontaneous malo. There's very, very little control over the microflora of the wine. So that leads to a variable speed of malo. It could go through really quickly. And if it's above pH 3.5, we often see Pediococcus or lactobacilli present, which can be very, very fast. Below pH 3.5, it will just be Oenococcus, which will be much slower. So we see variable speed of MLF. And if we do have a long delay between primary fermentation and malo, you could have wine that's warm, that's been unsulfured, which really could open the door for undesirable bacteria and yeasts. And actually inoculating for malolactic is one of the most effective strategies, combined with other strategies, of course, but one of the most effective strategies for controlling Brettanomyces. And because of that, we've seen in many of the traditional regions in Europe actually a very, very strong shift away from spontaneous towards inoculating. So just to change tack slightly for the next few slides, we thought it'd be really interesting to look at what we do as a company and where we get our strains from and how we develop the bacteria that we sell to yourselves as winemakers. So where do our malo strains come from and where do they originate? Well, we have an ongoing program in Christian Hansen, and the team that I'm going to be joining at the end of this month is the team that's responsible for commercializing our wine cultures. And we've got an ongoing program where we're always screening for what we would say is the next robust bacteria. Now, the 4 things we look for when we screen is first of all robustness in extreme and changing conditions. So it could be strains that can tolerate lower levels of nitrogen, or it could be high levels of alcohol, but strains that are going to be really able to tolerate extreme conditions, and maybe not just one. We at the moment we have a few blockbuster strains that have one really strong facet. Maybe trying to find a strain that performs well, both perhaps say with high alcohol and low nitrogen and low pH. If we could do that, that would be fantastic. Flavors are a really important thing as well, and it's an area where the science of malolactics really shifted away from a simple acid conversion, which is still the underlying process, to actually the impact that they can have on flavor. And with that, malolactic bacteria started to become a little bit of a stylistic tool. For instance, if you want to really produce a buttery Chardonnay, we would suggest a relatively slow strain such as CH35. It's a very slow but robust strain. That really allows the winemaking conditions to produce the maximum level of diacetyl. If you wanted to actually minimize the diacetyl, then using a strain such as the Sene, which is citrate negative, it's unable to produce the diacetyl, is really great for absolutely pure fruit expression. Or it could be, we've been working with some Lactobacilli plantarum recently, which produce a compound called ethyl lactate, which gives a very bright strawberry character. So in a red wine put through malo with this strain, it tends to have a really bright red fruit character. The third point is biological protection. As I mentioned, inoculating with Oenococcus is one of the most effective strategies against Brettanomyces. But we've also got strains of malolactic bacteria that can be dosed into grapes, machine harvested fruit when you're shipping across large distances, for instance. And then lastly, obviously improving quality and efficiency of production is important for us as a company. Christian Hansen is the largest producer of lactic acid bacteria in the world, and we have a huge production and technological platform that works across multiple industries. And us in wine, we're able to piggyback on that to make bacteria even more efficiently and even better adapted to winemaking conditions. All this begs the question, where do we actually get the strains from? Well, we do not use any genetic modification. All of our strains are isolated from nature, and they are isolated either from a grape, must, or winery environment. It's really important. We have activities in quite a few countries that are members of OIV, and the OIV rules do stipulate that you can only commercialize strains taken from those from a winery or winemaking environment. So we respect that, of course. We have a very large collection internally of strains. So when we are screening, we often go back to our own collection to see, is there an important character that we could use from our own strains we already own? We also collaborate with universities and research institutes, and it could be commissioning them to do a project for us to isolate something, or it could simply be asking if we can screen some of their strains as well. So I'm going to go on to this next slide, and this is similar to the slide that David introduced. But the key message we wanted to put across with this is that we have a range of 6 strains, and that each strain is different and has different characteristics and a different niche, if you like. So all of our strains are selected according to their different strengths. And with this range of 6, we really feel we can cover the vast majority of wines in the world that will go through malolactic. Just to talk a little bit more about the flavor, I mentioned that on the last slide. If we look at something like CH35, I'm just going to bring up my pointer. So CH35 here, it tends to give a very, very buttery character. So it's perfect for a traditional barrel or Chardonnay, where you want that buttery hit. If you actually want the antithesis of that, then we'd recommend our citrate negative, which produces absolutely no diacetyl at all. If you have a high alcohol, CH16 is really strong at, we say up to 16. We actually see it work in wines even above that from time to time. Whereas if you're looking at low pH, a strain like CH11 is a perfect one to use. So at this point, I want to introduce something called the magic number. And the magic number is something we use, but the magic number is a marketing term, but it's actually based on science. And it's in the scientific community, it's been well proven that the critical concentration of Oenococcus cells required for malolactic to happen is 1 million cells per mil, or 10 to the power of 6. So this is just to highlight that where our cultures are positioned. If we have this X-axis on the left-hand side, we've called that, that's the cell count, and we have M represented as a magic number. We have this here. All of our VINIFLORA bacterial cultures have a minimum specification of 2.4 times the magic number. So they're based on a very, very high cell count. Along the bottom here, this represents culture activity, which is also based on adaptation of the cells. And we have an internal test called MAC, which is malic acid conversion capacity. And all of our cells have very, very high activity and very, very good adaptation to wine conditions. Now, to try and to sort of put a case study behind this, we have some really interesting data from Australia. Now, this was run 15 years ago, but it's still extremely relevant today. And we've had a wine that's been put through primary fermentation, and then it's split into 3 different tanks. And this is part of a very, very big project, which our former colleague Peter Summer did when he worked for Christian Hansen. It's a very, very wide project where we're actually validating a new a frozen format for our VINIFLORA bacteria. Before that, we only had freeze-dried. So this is part of a big study, but this is really, really interesting because it highlights a few key points really, really well. So the top tank we had, so the wine was fermented, split into 3 different treatments, and at the top we have the one which was directly inoculated with VINIFLORA enos. a couple of really interesting points. First of all, the dark blue line is the bacterial cell count. And as you can see, that's sitting at around 3 times 10 to the power of 6. So we have the specification of 2.4 at the end of life. So about 3 is what you would typically see in the wine, and we're seeing all of the cells survive. That's the first point. And actually thrive. And we can see over time the cell count does slightly increase. The other really good thing we see is the light blue line represents the malic acid, and the malic acid starts to drop pretty much straight away. And it's a really nice clean malo, and it's down to 0.2, which at the winery, this winery, 0.2 grams per litre of malic acid was their specification for malo to complete. And in 20 days, the tank's finished malo. So it's really effective. And this wine graded really well. It was bright, it was clean. The next treatment we had was a cross-seeded tank. So this was where a tank that had already been inoculated with VINIFLORA enos and was halfway through malo was cross-seeded at a low percentage into the cross-seeded treatment. Now, what's really interesting here is we see a cell count in the early 10 to the power of 5. And that's roughly what we'd see if 5% by volume is cross-inoculated. That's the sort of level you'd see. And if they're cells that are surviving and they're growing up slowly, but they're taking around 13 to 14 days before they actually hit the magic number. And we've highlighted the magic number on all of these graphs by this red dotted line, which is 1 million cells. And then once it hits the magic number, we start to see the malic acid drop. So this does really highlight the importance of the magic number. And this wine completed malo in around 35 days. So around 15 days after the direct inoculated one. At the time, the winery where this work was done, cross-seeding was their standard practice. So it was a really interesting treatment to look at. However, there was a shift towards direct inoculation based on these results. Lastly, we also did one with spontaneous, and we have the dark blue line, which is the cell count, and as you can see, it starts off very, very low. The malic acid, the light blue line, still very high, and even after 30 days, the treatment with spontaneous still only has around 10 to the power of 4 CFUs per mil. So that tank's not going to go through malo for another few weeks, just while that population builds up slowly. Meanwhile, this wine's warm and unsulfured, which really does open the door for spoilage organisms to also build up. So that's the magic number. I'm now going to move on to a little bit about quality control. We have extensive quality control in Christian Hansen. We supply bacteria to pretty much every industry that uses lactic acid bacteria, and it's absolutely key that we have the utmost quality control. One thing where it all starts, one of the big risks for the population globally when we're looking at cultures is antibiotic resistance. So we make sure that all the strains we do commercialize do not have antibiotic resistance. And the second biggest area that we screen for is the production of biogenic amines. So before we commercialize any strain or before we even make a shortlist, this is screened for. Now, biogenic amines you may or may not have come across before. They're amines that are often naturally present in wine. But the process, the winemaking process, which leads to the highest amount, is generally malolactic fermentation. Now, the most abundant biogenic amine we see is histamine. And that's really interesting. All of the biogenic amines are produced by a decarboxylation pathway that some bacteria have. And we've represented this at the bottom here, where we have a histidine molecule. It's an amino acid, so it's naturally present in juices. And if you have any bacteria present with a decarboxylase enzyme, they will then produce histamine. Now, all of our VINIFLORA bacteria are screened and do not have this pathway, so they are unable to produce biogenic amines. And that's really important, because by inoculating and relying on strains that are unable to produce histamine, that's the most effective measure that can be used. Now, it's also really interesting with histamine. Some countries do use it as a trade barrier. We see that from the provincial monopolies in Canada and some of the monopolies in Scandinavia. And Switzerland as well did have a law which has actually been suspended at the moment because they want to harmonise with the EU, which just has a guideline. But nevertheless, there is legislation in place that does stipulate that biogenic amines do need to be controlled, but the most effective way to control them is to inoculate with a strain, so you have malo that doesn't result in that. Just on a similar theme, with Christian Hansen, we can supply the documentation needed, both for HACCP and for food safety modernisation processes, to help back up that, to help meet auditors' requests and to be aligned with modern food safety. So that's different product documentation, inoculation protocols, safety sheets, specifications, certificates of analysis, allergens, non-GM, all of those things. And a couple of further points: all of our bacteria cultures that we sell through Gusmer are validated for organic wines. As I mentioned, all the cultures are biogenic, amine negative, and we can supply certificates or documentation to back that up as well, which is important. At this point, just want to move on to inoculation protocols. And timing of inoculation has become something that we see is quite an interesting winemaking tool. So traditionally, whether it was inoculated or non-inoculated, you would typically see this process here with fecal sequential inoculation, where your yeast would drive the primary fermentation, and then a bacterial population would drive the malo. And David had that population graph at the introduction, which helps highlight that. Nowadays, we do see a lot more co-inoculation around the world, and that's where you start to have an overlap between your yeast population and your bacteria population, and therefore your primary fermentation and your malolactic. Now, early co-inoculation is where you would have both populations speaking together. This is where you would typically add bacteria around 24 hours after you would add yeast. And we also have late co-inoculation, which is where you would add your bacteria in the tail of primary fermentation, probably around 3 or 4 bricks. They're very, very good tools. They're not necessarily suitable for every wine, but nevertheless, they're quite interesting. Then we have sequential, which would be your traditional way, where we use yeast, and if it's a red wine, you'd press off, you would go to sugar dryness, and then you would have your bacteria. And we also see delayed inoculation, particularly for Pinot Noir. And this is where the colour stabilisation from allowing the wine to sit over winter and anthocyanins to polymerize with other polyphenols to help stabilize the color at a low pH would occur, and then you would have malo in the spring. We don't necessarily recommend this process because of the risk of contamination. If you've got wine sitting there in barrel with no sulfur, that is a Brettanomyces risk. However, if spring malo is what a winemaker wants to do, what we strongly recommend is to take the wine through primary fermentation. Once it's sugar dry, inoculate with bacteria straight away and keep the wine cool. And the really good thing with the bacteria is that it won't die. It's sitting there cold over winter in cold wine. Once spring arrives and the wine starts to warm up naturally, you've got an active population that just needs a little bit of heat to complete the malo. And you've also got the protection of the dominant population there over winter. So that's what we'd recommend for spring malo or delayed inoculation. And just one last point, reverse inoculation is something we've been playing around with, and that's actually doing partial or full malo before you actually inoculate with yeast. Now, we don't recommend this with Oenococcus. I've seen some interesting data from Germany where it's been done. And it's been used to take 1 gram or 2 of malic acid out of the wine before yeast. It needs to be very, very tightly controlled. Or if you use it using something like a Lactobacillus plantarum, it can also be used because Lactobacillus plantarum doesn't produce any acetic acid from sugar. So it's an interesting one. We'll see more and more of reverse inoculation in the future, I believe. So I just want to talk about some of the benefits of co-inoculation, particularly early co-inoculation. It is a technique that we're seeing growing popularity with. For instance, in Australia, where I've been based for the last 5 years, I would estimate that around 70% of the bacteria that we supply is used with some form of co-inoculation. And what's really driven it is the larger winemaking groups have seen the benefits and have the control and the modern facilities to be able to control it, and have really adopted it and embraced it in a big way, and have seen some really big benefits. So what are these benefits? First of all, in time, it's less time. The winemaking process is effectively shortened. You could be pressing your wine off after 2 weeks, and it's basically both sugar dry and malo dry, which is amazing. It can then go on to further blending or finishing. It's interesting. One of the earliest adopters of early co-inoculation was for Beaujolais Nouveau, simply because the wine has to be released in November, so it has to be fermented quickly. There's cost savings often with co-inoculation. If you don't have to heat the wine for malo because it's already gone through, that's quite a good saving. Also, it reduces the carbon footprint. Also gives very good control. And if you're looking at a wine that has particularly high brix or high sugar, or is at a high level of extraction or high level of polyphenols, co-inoculation is a very good way of addressing those wines, because both the phenolics and the alcohol can be inhibitory. So by introducing the bacteria early, before those inhibitory factors have reached their maximum concentration, you're getting a population already fermenting away and getting well adapted to the wine, which will help it go through once you have finished your extraction or your fermentation. So what are the impacts on the final wine? Co-inoculation is a really good way to avoid spoilage, because you're able to sulfur the wine and filter it much sooner. And then lastly, it actually tends to enhance fruitiness in red wines, and that's because Malolactic bacteria will produce diacetyl. But when they produce diacetyl and yeast is present, the viable yeast cells will actually consume some of that diacetyl, which tends to give you very bright, fruity wines. So this is where co-inoculation becomes somewhat of a stylistic tool. So it's a very effective technique. It's also fair to say that sequential fermentation very much has its place, but the choice is there, and it comes down to what fits in with the wine style and what fits in with the cellar. So for the next few slides, I just want to look at troubleshooting and stuck fermentation. It's one area with my role in Australia I've been heavily involved with, talking to our customer base every year, usually around May or June, once the vintage is over, when there's stuck tanks. And so it's something we've been heavily involved in, and I think it's absolutely no different with Gusmer in the US. The Gusmer reps come across stuck fermentation all the time and can advise on the best ways to solve those issues. But first of all, if we were to take you through a sort of thought process with dealing with a stuck malo, first of all, it's good to look at the basic parameters: the pH, the alcohol, the sulfur, and the temperature. I'll just make a comment about sulfur. When we talk about sulfur and bacteria, we talk about totals, not free or molecular. Now, the reason we do that is sulfur dioxide, when it's bound, whether it's bound to maybe an aldehyde or something else in the wine, can still be brought across the cell wall, so it's still toxic. So we tend to talk about total sulfur as being the important parameter for that very reason. So the basic parameters are the ones to look at, and often by choosing a more appropriate strain, perhaps you might have an alcohol of 15.5%. You can solve, but you've inoculated with a strain that doesn't have high alcohol tolerance. Just by simply inoculating with a more appropriate strain, it can solve the problem. that's the first thing to check. The second area is nutrition, or the nitrogen. Now, MLF does require 60 parts per million of amino nitrogen to proceed. So for that, we would actually recommend re-inoculation with a malonutrient if you do have a stuck tank. Now, malonutrients are usually yeast derivatives, and they provide amino acid in the form of small yeast peptides, which is actually the preferential format for Oenococcus. Malonutrients will also bind medium chain fatty acids. So for certain styles, we actually recommend using a malonutrient just as part of the standard ad, particularly if you're using a champagne style yeast, just because they will bind medium chain fatty acids. Plus, the champagne style yeast tend to be quite greedy on nitrogen, so the malonutrient will put back the amino nitrogen required for malo. I'll just also make a comment with nitrogen. You can measure the amino nitrogen. An enzymatic test kit that can measure amino acids is what we generally recommend if it's something you wanted to measure. Sometimes it's just easier to add a malonutrient, and so that's the best route, but it can be measured as well. The third point we've got on here is the cell count, CFU per mil. It's often good to see how many Oenococcus cells are in the wine. One thing we do see from customers that if there's a stuck ferment, the first step intuitively is to add bacteria. But sometimes we've actually analysed stuck tanks, and there's plenty of Oenococcus cells already there. So re-inoculating is going to do nothing. It's just that there's something in the environment stopping the population already inoculated from fermenting. So that could be low nitrogen, or it could be actually phenolics. We've seen some really interesting and sometimes worrying results with certain varieties. We see Merlot is probably the worst, and by that I mean not sort of commercial Merlot, but pretty high-end Merlot with a lot of extraction can be a really difficult wine to get through Malo. And there's certain phenolic compounds that Merlot happens to have in abundance, which tend to be very inhibitory to malolactic bacteria. So phenolics can be a big issue. For wines like that, we do recommend co-inoculation, especially late co-inoculation, because it's a way of getting the bacteria established before the extraction is finished, particularly if there's a post-ferment maceration. And we've also seen some really interesting results from, excuse me, from Italy, finding with PVPP to reduce the phenolic concentration slightly, and then allowing Malo to proceed. And that's an interesting tool. It's not something that you would rush into. Obviously, it would require a finding trial on the bench first, but nevertheless, it is a tool that can be used to help to get a wine through Malo if it is phenolic inhibition. And the other option we do see from time to time, and it's less ideal, but particularly if you are looking to do a blend later, actually do your blending before Malo. If it's Merlot you're dealing with other varieties, that way you can mitigate against the phenolics in that variety.
Duncan, we have a question regarding cell counts.
Yep.
And that is, if I add CH16 at 3 to 4 bricks, do I use the must volume or the expected post-press volume for calculating the amount of bacteria to add?
I would use the post-press volume, the liquid phase only, because the skin's there, they're solid, but it's the liquid where the bacteria are really going to be living and surviving. So I'd go by just the liquid. That's a really good question.
All right, thank you, Duncan.
No worries. And if I can make another comment on that, if you are inoculating at that sort of level, look, at 3 to 4 bricks, you're going to be having a cap. It's really important to get the bacteria into the liquid at the bottom and not into the cap. The cap's going to be warm and it's going to be very rich in phenolics, which are inhibitory. So it's really important to either have a pipe or something to break through the cap, or to set up a pump that's going straight into the liquid phase, or worst, inoculating during a pump over, just to make sure that the bacteria is not staying in the cap that goes into the liquid phase. So this is my second to last slide, and it's just to reiterate what we've already talked about. Wine is a harsh environment for any microbe, but Oenococcus is a pretty hardy organism and can survive it. Nevertheless, we do see times where it gets too harsh. So I've just tried to make a summary table here on the left of what we've already talked about in terms of the different inhibitory factors: the pH, the temperature, the malic acid, the SO2, and the amino nitrogen, to just try and put some numbers between what's easy and what's difficult. Sometimes pre-malo, it's important not just to select the right strain, but sometimes the wine conditions do need to be adapted to fit a strain at all. However, in Gusmer, you have an extremely capable and knowledgeable resource. So if you do have questions about what strain to use, we would highly recommend talking to your local Gusmer rep to take their advice. So this is my last slide, and I'm not going to talk through all of this. This is the range of bacteria that Gusmer supply, of Christian Hansen bacteria that Gusmer supply into North America. We believe we have a strain for pretty much all wines that need to go through malo in North America. And we'd also say it's really important to try and choose the strain that fits the winemaking conditions. And on that note, I think we'll see if there's any more questions.
As we're waiting for questions, I just want to remind everybody that we will send out a link to this, the recording of this presentation, but also want to say thank you to everybody who attended. We know you're busy. Thank you for taking the time. David, Duncan, thank you so much for your partnership and taking the time for the presentation today. Thank you.
Cool. Thanks, Chris.
Okay, so we have a couple. Is Gusmer experimenting with breeding bacteria, as many companies are doing with yeast? And so Christian Hansen, partner of Gusmer, is the one that is producing and supplying the yeast and bacteria. But Duncan, do you want to say anything on breeding bacteria? I know you touched on it briefly about how we go out and find the bacteria in the wine industry.
Yeah, thanks, David. I would say in terms of breeding, not so much, because the rules that we stick to with commercializing strains really do stipulate they have to be natural isolates. That isn't to say that we can't utilize mutations that the natural isolate might exhibit, but generally speaking, with bacteria, we can't do breeding like you can with maybe doing hybridized yeast and things like that. So the far easiest way is to actually screen the collections of bacteria that already exist for the traits that you want. And then at the other end, where we can work as a company is actually in the production, but that's not doing anything to the actual strain itself. It's how we can grow it internally.
All right, thank you, Duncan. The next question is, how many grams per hectoliter do you recommend to add to red ferments? And I think maybe briefly you can just touch on how we deliver our bacteria, in terms of not per gram, but per wine to be inoculated.
Exactly, that's right. Yeah. So Eric, so the way that we package our bacteria, they'll be in a packet, and it'll either be for 250 liters. David, you're going to have to help me with the gallons. That's about 50 gallons, right? And then 2,500 liters or 2,000 or 25,000 liters. So we stipulate the amount on the packet that it should go into, and there's a really good reason for that. All of our cultures are pure culture. There's no filler. And so when we do produce cultures commercially, we do see a natural variation in the mass produced. And what we do is when we sell it, the specifications are all around the cell numbers. So we keep the cell numbers the same batch to batch, but the actual amount of physical material can vary. Based on that, we don't have a grams per hectoliter. We tend to label a pack size to deliver a minimum of 2.4 million each time you inoculate. That's also really important because all of our cultures are suitable for organics. If we were to use a filler, we believe that organic approval would become a bit more of a grey area because we're no longer working with pure cultures. So as a company, we're very tightly aligned to a dose rate stated on a pack.
Okay.
Would you add anything, David, to that?
That's a good answer. This is what I'm going to call the lightning round because we only have 2 minutes left.
Oh, really?
2 questions. So I'm going to read both questions and then you have less than 2 minutes to answer them. Okay. Can you comment briefly about reverse inoculation? Is that some new trends and what bacteria is having the more participation in this process? And he says, one says, great presentation. Second question, does the MLF bacteria that Gusmer carries all for co-inoculation, or is there a specific product that is great for co-inoculation? And then there's a couple more questions. I'll write those down and we'll get back to them at another time. But can you just answer those first 2 questions?
I'd say that reverse inoculation's not quite become a trend. It's still become it's something that's been trialed, but it hasn't been adopted as a wide as a broad trend as yet. I've seen it both with Oenococcus and with Lactobacillus plantarum. Plantarum's more suitable, but it's not suitable in every market either. So I think we'll see more in the future, but at the moment, the big risk as well with Oenococcus is that you can get high levels of VA if you don't control it properly. So on that, we don't recommend it. Next question, all of our bacteria are suitable for co-inoculation, early co-inoculation, late co-inoculation, or sequential. We don't distinguish between them, and they're all suitable for all 3 approaches, which is really strong. A lot of companies have products just for co-inoculation, which will absolutely fall over if you use them sequentially. All of ours are robust enough to do any of them. Do we have one more? Phenolics? Good question about phenolics. I think we're still working out exactly about the phenolics. To be really honest, it depends on the variety, and it's not really been as well studied. There was a really good paper out of I think it was from Spain about 10 years ago, and we've seen very little academically since. So it tends to be quite anecdotal, our experience on it. But watch the space. We'll do more in the future. Yeah. Does that help, David?
Yes, it does. Thank you very much, Duncan. We really appreciate your time and looking forward to maybe seeing you at the Gusmer booth at the 2022 Unified Symposium.
Yes, that'd be great. That'd be absolutely awesome.
Thank you again, Duncan and David. And anybody else, if you have questions, feel free to reach out to your sales rep or reach out to Gusmer directly. Happy to help. So thank you.
Cool.